Bismillah ar-Rahman ar-Rahim. Assalamu alaikum. I am Professor Adam Ahmad Adam. You are welcome to this parasitology lecture. It is about malaria. I divided this topic into 3 parts. This is part 1. We start part 1. Malaria is a fever disease caused by the protozoan parasites Plasmodium species. The Plasmodium species that cause malaria are 1. Plasmodium falciparum, 2. Plasmodium vivax, 3. Plasmodium ovale, 4. Plasmodium malariae, and 5. Plasmodium knowlesi. I would like to say a few words about Plasmodium knowlesi as the cause of malaria because it is a newly discovered plasmodium to cause human malaria. It is found in Southeast Asia, especially Malaysia. It is a zoonotic parasite because it infects non-human primates, long-tailed macaque and pig-tailed macaque. You see the pictures. Plasmodium species have 2 types of life cycle. One asexual life cycle, which is known as schizogony. It takes place in human. 2. Sexual life cycle, known as sporogony. It occurs in the female Anopheles mosquito, which is the biological vector of malaria. Most of infection, how the infection takes place. 1. By the bite of the infected female Anopheles mosquito, and this is the main mode of infection. This mosquito is the biological vector. That is to say, where the parasite lives part of its life cycle. Example of Anopheles species: Anopheles arabiensis, Anopheles gambii, Anopheles stephensii, and the list is long. The infected female Anopheles mosquito bites human, mainly nocturnal, that is to say during the night, to take a blood meal for egg production. So it needs blood to produce eggs. The male mosquito does not feed on blood, it feeds on rose nectar. It injects the sporozoites, the infective stages that are found in its saliva, into human blood circulation. So the infected female Anopheles mosquito, during biting human, injects the sporozoites into human blood circulation. You see here, this is the picture of the female Anopheles mosquito and also the infective stage, the sporozoid. The second mode of transmission is blood transfusion. The infective stages here are the asexual erythrocytic blood stages, the stages that are found in red blood cells. If a person is transfused with blood from a donor with malaria, he or she may contract malaria from this blood. I would like to mention here that Plasmodium species can remain viable alive in refrigerated blood for about 2 weeks. The third mode of transmission is transplacental, through the placenta, also known as congenital. The pregnant woman with malaria can infect her baby in the uterus. The parasites pass from the mother blood to the baby through the placenta. The child may be born with malaria. The infective stages here are the asexual erythrocytic blood stages, the stages in the red blood cells. Here we come to the sequence of events after injection of the sporozoite into human blood. So you can follow the numbering. The sporozoites, after being injected in the blood, they circulate in the circulation for about half an hour. Then they enter the hepatocytes, the liver cells. Each sporozoite forms a vacuole in the hepatocyte known as the parasitophorous vacuole, and it becomes a trophozoite feeding and growing stage that develops into schizoid, the dividing stage. In the schizoid stage, the parasite divides to form merozoites. After full maturation of the schizoid, the hepatocyte ruptures, releasing merozoites that invade the blood and infect the red blood cells. I would like to mention here that in case of Plasmodium vivax and Plasmodium ovale, some of their sporozoites, known as hypnozoites, remain dormant in the liver cells and wake up later in time, maybe weeks, months, or even years. So they do not develop immediately into the trophozoite and schizoid. They remain sleeping there. So they are known as hypnozoites. Later, they may wake up and lead to malaria relapse. After rupturing of the infected liver cell, the tissue or exoerythrocytic merozoites emerge from the ruptured hepatocyte and infect the red blood cells. Red blood cell infection is preferential. That is to say, each species of Plasmodium prefers certain age of red blood cells. Here, Plasmodium falciparum infects all ages of red blood cells. Their merozoites infect all ages of red blood cells. Plasmodium malariae, the merozoites infect all red blood cells. But in case of Plasmodium vivax and Plasmodium ovale, they infect the young red blood cells, which are the reticulocytes. And we see here that the red blood cells at all ages are targets for Plasmodium falciparum, and this is why the parasitemia is high in Plasmodium malaria compared to other types of malaria. Here inside the red blood cell, most of the merozoites develop into asexual stages. You follow the numbering, the merozoite changes into ring stage, then into trophozoite, then schizoid. After full maturation of the schizoid, the infected red blood cell ruptures, releasing merozoites and hemozoin. Hemozoin is the byproduct of hemoglobin digestion by the parasite. It mediates symptoms and signs of malaria. The emerging merozoites reinfect other healthy red blood cells and the cycle continues. Follow the figures, numbering and the arrows. Some of the merozoites that infect the red blood cells develop into gametocytes, which are the sexual stages. If these gametocytes are sucked in a blood meal by a female Anopheles mosquito, the sexual life cycle, which is sporogamy, starts. The gametocytes themselves do not cause clinical malaria, but they infect the female Anopheles mosquito to continue parasite transmission. There are male gametocytes known as microgametes and female gametocytes known as macrogametes. By this part is over. I hope that it is understandable. Please proceed to part 2 and I wish you all the best.